Hello and welcome to our last part of the history and introduction to microbiology. So we are going to look at immunity, chemotherapy, and infection prevention. We start off with Edward Jenner, who said the artificial inoculation with an agent of reduced virulence when introduced into a man will produce a mild form of the infection. And he also said that this will give protection against the natural disease. For example, if you give a weakened form of hepatitis virus into a man, he says it's not going to cause the whole disease in a man, it's rather going to cause a milder form of the infection. However, the person is also going to receive protection against the disease should the person catch the disease later on in life. This was the beginning of immunization. And so, what Edward Jenner did was he vaccinated people against smallpox, but then he used another disease which was cowpox. Now, he had heard a milkmaid who said that I will never get the smallpox because I have already had the cowpox. So Edward Jenner decided to vaccinate an 8-year-old boy with the cowpox, which had been derived from a milkmaid. So the boy developed milder symptoms of the cowpox after some time. Now, few months later, Edward Jenner vaccinated this same boy again, but this time around with the regular smallpox. But the boy did not develop any symptoms at all of the smallpox. And so Edward Jenner then drew the conclusion that the boy was completely protected from smallpox due to the cowpox which he was vaccinated earlier. And so after repeating this procedure several times, he called this process vaccination, after the Latin word for cowpox, vaccinia. And so vaccination against smallpox eventually led the World Health Organization to declare the world smallpox free in 1980 and stop vaccinating against it worldwide. And so the injection of cowpox, which was vaccinia, was given to protect against the smallpox, which is also known as variola. And so after this, Louis Pasteur and others also showed that this principle could be applied to other infections. And so Salmon and others demonstrated the use of the killed organism to also generate, some form of immunity in man. Now, after this, we had Karl Landsteiner, who studied the nature of antigen and antibody reaction in the body. And he was the one who said that there are 4 main types of blood groups. And he said we had the blood group A, B, AB, and O. Now this led to the development of diagnostic serology by Georges and Fernand as well as Woodall et al. But it was Erlich who described cellular immunity and said phagocytosis is a cell-mediated immune response to foreign matter. But prior to this, it was believed that white blood cells, for example, were the ones which take up the bacteria and spread it throughout the body. But now we've gotten to know that it is rather the white blood cells which fight against the bacteria. And phagocytosis is part of the activities of the white blood cell. Then we had the famous Alexander Fleming, who in 1928 observed that growth of the bacterium Staphylococcus aureus was inhibited in the areas surrounded by the colony of mold that had contaminated a petri dish. So what he realized was that he was growing the bacterium Staphylococcus aureus in the lab, but then he realized that some parts or some of the microorganisms were not growing at all. And this was the part of the dish which had been contaminated by a mold over here. So he realized that it was the mold which was preventing the growth of the Staphylococcus aureus over here. So what did he do? So he identified the mold as Penicillium notatum, and this later on became known as penicillin, and it was the first antibiotic that was made. Now, in 1942, injections of the penicillin had been prepared, and it was used for treatment in humans. Then later on, we had actinomycin also being produced. Now, another scientist was Paul Erlich, who was a German chemist, and he noticed that dyes were selectively absorbed in some cells, and these could selectively destroy bacteria but not animal cells, which we now use as chemotherapy. Now, he developed some chemical compounds, and we had, the asphenamine, and also 606, or the salvarsan, which was used to treat syphilis. Now, in 1890, he proposed a theory of immunity in which antibodies were responsible for immunity, and he is known as the father of modern chemotherapy. So he proposed the magic bullet of selective toxicity, and what he stated was that if a compound could be made that selectively targeted a disease-causing organism, then a toxin for that organism could be delivered along with the agent of selectivity, hence the term selective toxicity. He also developed staining procedures to identify the tubercle bacilli, a method which later on was modified by Zhou and Nelson, which is still used today and subsequently developed into gram staining. Then we have Joseph Lester, who controlled infection in the operating rooms by the use of phenol. So he sprayed the air around the patient with phenol to prevent, infection in the operating room, and also surgical site infection. He also, controlled the spread of infection from surgeons to their patients, and so this that he brought helped decrease surgical mortality in the operating rooms, and also aesthetic surgery was developed later on from there. Then we have Ignace Semmelweis, who noticed that women in the labor ward were infected after being examined by physicians who came out directly from the autopsy room. He therefore advocated for hand washing between patients and among health professionals. Now, this helped to decrease the incidence of septicemia after childbirth from 35% to 1%. And so it was this scientist who actually first introduced hand washing. Then we have Jon Snow, who in 1854 noted during a cholera pandemic, or a cholera epidemic, that water was contaminated with human feces and could cause the disease cholera. He said this was due to the lack of public toilets, improper drainage, and also stream water, which was used for drinking. And so we began building public toilets, and also constructing proper drains, and also educating people to stop drinking from stream water. Then finally, we had viruses, and before the 1930s, any living agent, if disease, including bacteria, was thought to be a virus. Then we later on got to know that we could filter some agents, which passed through special filters. But in 1940, we were able to determine that extremely small non-cellular particles which infect and replicate only within living cells, to be viruses. Now, after that, we had several, plant viruses which were described, and we also had a strain of the polio virus which was seen, and we also used lots of tissues to culture, viruses, and we also used, eggs to culture viruses. Now, we also need to know about the classification of living things and also the domains of life. And so we have for the bacteria, we have the archaea, then we have the eukarya. And so when we look at the domain bacteria, we have the eubacteria, which is the true bacteria. The domain archaea, we have the primitive bacteria. Then the domain eukarya, we have the protozoa, fungi, plant, and animal. This we shall look into detail in our class. Thank you very much for your time. Bye bye.